I'm Alastair McGinness. I'm a third-year PhD student at Oxford Brookes. My supervisor is the lovely Verena Kriechbaumer, and I'm doing a really nice project which is modifying N-linked glycosylation in plants in order to produce human therapeutics. I'm mostly looking at, when I talk about N-linked glycosylation, really what it's a discussion of is how to make drugs, medicines, biological products that are of use safer for humans to use. Because what N-linked glycosylation is, it's a very specific addition of sugars onto finished product that's produced in cells. And what that's responsible for is it does a lot to say, where has that product come from? Where has it been synthesized? Does it look, in this case, does it look human? Is my major concern. And we're trying to find ways that we can achieve that in plants so that, for instance, you take a therapeutic, it looks to the immune system like it came from a human body. And then your body goes, oh, I don't need to get rid of that. It's not a threat. I'm not going to have an immune reaction to it. So it's safer. Basically, you get very, very specific post-translational modifications. So these kind of hallmarks of where something's been produced between different species, between different clades of life. And what I'm basically trying to do is there's very specific human glycosylation, so sugar additions. There's a specific one which we're trying to do called N-terminal sialic acid. So it builds, it's the final step on a long chain of sugars. And it's really key to identifying something came from the human immune system. And it's a key mitigator of that. For instance, the COVID S spike protein, where there's all that discussion about, is covered in terminal sialic acids, because that's what it uses to evade the immune system. Because that's what's normally produced in a human, that's what our proteins are covered in. So by manipulating, for instance, I'm manipulating plants, I'm putting human enzymes to produce that sugar into the plant Golgi, where the post-translational modification occurs, and that way anything that's been synthesized in that plant has that terminal sialic acid, and to the human immune system goes, oh, that was produced here, that's safe.
Awesome. And so, yeah, so I'm interested. So being part of the microscopy group. I'm assuming you do a lot of cool stuff with microscopes. And could you talk us through, yeah, a bit of like your microscope work and like the actual like what you do, how you do this? Talk us through the methodology.
Yeah, so I mean, I wouldn't say basically, but it's not that basic. It's lovely though. A lot of what I do is focused on, when it comes to microscopy, we're really testing about where are things going. If you want to express something in a plant, so if you want to get a gene, you want to get it working in a plant, you want to produce a product, you want to do any of these things, you need to know where it localizes. And that just means, where is it going in the plant? And the most effective way to do that is you get the microscope, you get whatever you're trying to implement, the plant, the gene, and you fuse a fluorescent protein to it. So that when you look at it on the microscope, you've got this bright fluorescent color, so you can go, oh, that's where that's going. And so a lot of my work actually is I'm looking at the plant Golgi, which is the major site of previously discussed post-translational modification, the sugar addition. I'm getting these human sugar addition enzymes, inserting them into the plant Golgi in very, very specific localization. They have to be in the late plant Golgi. And so I get different fluorescent proteins that go to different parts of the Golgi, and then I can look at the microscope and I can go, oh, comparing that fluorescent protein with that fluorescent protein, one I know that goes to the top, one I know goes to the bottom, we can sort of create a yardstick of where things are localizing within the Golgi. And so most of that I assess with a really nice microscope, which can do super resolution imaging. So below really the resolving power of light, you're talking the plant Golgi I'm looking at and looking at the dynamics of, so how it moves, where things are located, is like 200 nanometers across. It's tiny, tiny work, but it's great because I mean, I get to do sort of 10 hours of that a week on the microscope looking at these dynamics. It's fantastic. So most microscopes that people would use are light microscopes, which we'll sometimes call bright field because it's bright. So you get a white light, you shine it on a sample, and it's great for looking at lots of things. But you're able to see an image because light hits that object, and light basically diffracts or perturbates or something happens to it, and that way some of it is blocked, and you can see an image. And that's great, you can visualize a lot of different things. But now with the modern generation of microscopes, light's kind of a bit old hat. There's more we could be doing. So what I use is a confocal microscope, which uses lasers in order to, when you have a fluorescent protein, you can shine a laser on it to give it energy. and it takes in that energy in one wavelength. So you start with, let's say, blue light, you shine on your sample, and then it gives it at a completely different spectrum as green light. But a confocal microscope, we can see light has its limits. And specifically, we're thinking about how easy it is to resolve something, how closely can we image it, how precise can it be? And a lot of time we use lasers for that. So you can use a laser combined with a fluorescent protein, something like that, to give you a really high energy signal that's really nice and crisp. And what that lets you to do is it really pushes the limits of what you can actually image and how small you can go. So use of a confocal microscope is really great to, one of the major points is remove all the out of focus light. So we have this concept of kind of segmenting an image. You're looking at one plane in focus, and everything that's not above or below that is taken out of the image. So you get super crisp 2D slices of 3D structures. And one of the things we do a lot in our lab is this concept of you can build a 3D image by taking a series, we call it a Z-stack, of these 2D images to build up complex 3D structures and then reconstruct them on a computer as a 3D model that you can rotate and view the different angles of. And it completely revolutionized how we look at networks, how we look at cellular structures. And I just think it's such a cool concept I get to use almost on the daily. I think when it comes to like my career history, I think a lot of particularly undergraduates, people not in the sciences, have this idea that you start as a scientist with one goal, and that you have this very linear career progression of I want to study exactly this thing, this type of fish, and I'm going to go to undergrad and I'm going to do all the things I need to do exactly that. I'm going to go from there straight into a master's, straight into a PhD. And that, I would say generally, it hasn't been my experience. I had a really sort of lovely, long, meandering career. So I did my undergrad at the University of Surrey. I had a lovely time there doing an open-ended biological sciences course. And I got interested in a lot of different things. So I worked at a neurobiology research center doing sleep and then neuroinflammation studies during my undergrads. And there I sort of changed tracks because before I'd been studying neuroscience, I was convinced that's what I wanted to do. And I started doing the research environment in that. I was working in a lab in Germany and it just wasn't for me. So I sort of changed tracks, came back, final year dissertation, decided to do work more on natural product isolates, what you can get from the environment. It was looking at microbials in nature, particularly from lichens. And it just kind of opened up the world of bioproduction. What can we produce in organic systems? And I went from there. I think a non-traditional route a lot of students worry about is taking time out of academia. I actually worked as a chef for 2 years, doing my undergraduate and my postgraduate studies, and I honestly just don't regret it. It was fantastic perspective for coming back. But I really missed the research environment. I missed, I don't know, using my brain at work and actually engaging with biology. So I actually started my current project as a master's by research at Oxford Brookes. My supervisor, Verena, was incredible, getting to use confocal microscopy for the first time, like just really engaging with the world of plant sciences. And I just went, this is fantastic. And when my supervisor said, would you like to stay for a PhD? I thought, yeah, all right, sounds great. And there we are, I've been in Oxford for sort of 4 years now. I've got the final year of my PhD coming up. I'm Oxford BBSRC DTP, so I had a lovely 4-year program with a year of training and lab rotations at the start. And honestly, it's been a really nice kind of almost holistic journey with biology. I've got to explore so many things in my career so far that I thought were of interest to me and kind of pared down to what I'm really passionate about and what I want to do. So my PhD program is actually funded by the Biological and Technological Research Council, so that's the BBSRC, and then it's a doctoral training program. So quite often, and I really like this format, if you're not sold on a PhD title, but you know you want to do a PhD, you know you're really interested in research, this program in particular, you did sort of 3 months of really, really good stats, programming, microscopy, skills that most PhD students are really going to need, but won't necessarily learn until they absolutely have to. So it was a nice primer for that. But also, particularly for me, I got to do rotations in different labs with different PhD titles, with the concept of just exploring what that particular research climate feels like, whether it's a topic you're going to be interested in. And I mean, in the end, I ended up working with the same supervisor that I did for my master's because while I'd had a lovely time, it gave me the clarity to know exactly what I wanted to do. And so I'm still on the same program. I'll be finishing sometime next year, hopefully. And yeah, I'm having a fantastic time with it. It's hard to say what the best thing about what I do is because I think, not to be the person that I'm going to flip that question on its head, I get to do so much. There's so many things on a daily basis that I get to explore. For instance, just my workflow at the moment, I'm spending half my time in the microscope, half my time in the lab, where I'm regenerating lineages of stably transformed plants. It's exploring every research interest I had, and then getting to apply all these skills to them, getting to experience all these different techniques. And it's just nice to kind of have every day be a little bit different, be doing something a little bit new, be doing something a little bit fresh, and kind of synergizing those together onto one project is just a really nice research experience to have.